Hello everyone. We will start today's lecture. So, welcome to the week 2 Saturday session for novel technologies for food process and shelf life extension. So, in this live session again, we'll be solving some questions regarding week 2 syllabus. I'm aware that your assignment date has already been over. So, I'm guessing you will be familiar with the concepts that we'll be taking up in this particular session. But if you guys have any doubts, you can post it in the group chat or you can unmute yourself. If you guys want me to repeat any question as well, you can do the same. So we'll start with the first question. Which among the undermentioned option is correct for the following reaction process? So we have tyrosine as the substrate, and here you can see first it undergoes hydroxylation in the presence of oxygen and enzyme to yield a particular compound. That again undergoes oxidation to yield the second compound. So one first compound that you need to identify is this. The second compound that you need to identify is this particular molecule that also is formed upon oxidation of the first product. The third product is formed from the oxidation of the first product, and finally you see that there's a fourth product that is forming and then that contributes to the formation of melanin. So does this reaction look any familiar to you guys? What reaction are we looking at? The formation of melanin from tyrosine. Does this reaction ring a bell? Am I audible to you guys? If I'm audible, just post it in the group that you can hear me.
Browning reaction, ma'am.
Yeah, so this particular entire mechanism corresponds to the Browning reaction that is present in foods. So can you identify the first molecule here? What happens when you hydroxylate tyrosine? So here you can see one of the hydrogen present in the benzene core is being replaced with the help of replaced by a hydroxyl group. So what does two hydroxyl group and a phenylalanine group represent? So we'll move to the solution. The first reaction, that is tyrosine hydroxylated, in the enzyme that we're talking about is phenolase enzymes. These phenolase enzymes are present in food such as fruits like apples and all. These phenolase enzymes convert tyrosine to dihydroxy phenylalanine molecule. This is the phenylalanine moiety and this is the dihydroxy groups. So dihydroxy phenylalanine molecule is also known as DOPA. So the first compound that is produced from tyrosine is the DOPA or dihydroxy phenylalanine molecule. So the second reaction that we need to figure out is DOPA being converted into oxidized into some other molecule. Can you figure out what this molecule is? So what happens is that here it undergoes oxidation and the two hydrogen that are present in the hydroxyl group are oxidized to a keto form. So we're looking at DOPA is oxidized to dopoquinone. So when you have a benzene ring and two oxygen atoms attached in the by replacing the hydrogens, you have something called as a quinone molecule. So this is a dopaquinone molecule. The second molecule that we're looking at is dopaquinone. So the third reaction where DOPA is oxidized into some other molecule that has a specific ring like this sort. So what does this ring stands for? You have the same quinone-like molecule, and what is this? Any clue? What is the functional moiety here?
Amine, ma'am.
Sorry?
Amine group.
No, this is an amine group. What about the entire structure? So there is when there's a benzene core and a pentameric cyclic structure attached with an amine group, what is it called? What is the entire functional moiety called? So this is halochrome, and this is a precursor for the indole ring. So an indole ring will actually not have the COCH. Instead, it will have a benzene ring and a pentameric cyclic structure with an amine group. That is how an indole ring looks like. So what happens? This is the precursor for the indole group and is known as the halochrome red. So the third compound here, which is in red color. So this is the halochrome, and this is the third compound that is formed during the Browning reaction. So this is one of the first colors that is produced when tyrosine is being oxidized, undergoes oxidation and hydroxylation. So the precursor, this 5,6 dihydroxyindole is a molecule that comes after this molecule. So this is a precursor for this molecule. So what happens when this hydroxyindole undergoes oxidation? So this forms when 5,6-dihydroxyindole undergoes oxidation, it forms indole 5,6-quinone. Same, you have a benzene ring and 2 oxygen attached, and then you have an indole group. So this is indole 5,6-quinone. So basically, when you have a benzene ring and 2 oxygen atoms, it is called a quinone. When you have a pentameric cyclic structure attached to this benzene ring, it is known as indole quinone. So these indole quinones then lead on to produce what is known as melanin. And as we know, melanin gives a brown color, be it foods, be it skin, be it any tissues, it gives a brown color. And hence melanin and other melanin related compounds are the ones that are responsible for this browning reaction in foods. So this is a typical reaction of a browning process. The answer is option D. The first compound is dopa, second compound is dopaquinone, third compound is halochrome red, and fourth compound is indole 5,6-quinone. So moving to question 2. Which of the following statement is true about a protein? A. Proteins are of colloidal dimensions and as such they can pass through the semi-permeable membrane. B. In the polymeric forms, proteins possess an irregular specific folded three-dimensional conformation. C. Proteins can behave both as an acid and a base. And D. 21 amino acids make up food protein. So which of these statements is true?
Option C.
Yeah. So option C is true. So what about option A, B and D? So what is wrong in those statements? So proteins are of colloidal dimensions. They can be big enough, they can be extremely small, they can be big enough as well. But as such, proteins cannot pass through the semi-permeable membrane because first thing of their size, and second is that they have a lot of groups that will not, functional groups that will not allow it to pass through the membrane because membrane has a hydrophobic core as well. So the properties of the proteins and the size of the protein should be compatible with that of the semi-permeable membrane, which is not the case. So proteins cannot pass through the semi-permeable membrane. One of the reasons is because they are of colloidal dimensions. And option B is that in polymeric forms, protein possess a regular shape. So it does not have some random shape. It has a very specific folded and a regular shape. Where in case, like if you're looking at an alpha helix, there will be a hydrogen bond between the first residue and I plus 4, the 1 plus 4th residue. So you'll have a very specific arrangement of the hydrogen bonds and other interactions that will give it a very, very regular shape. So proteins do not have an irregular shape. That is what is false in the second statement. And C, which is correct, proteins can behave as both acid and a base. This is because proteins are made up of amino acids, like multiple amino acids form a protein, and each amino acid has an acidic group as well as a basic group. So this acidic group has the ability to donate hydrogen atoms, hence making it an acidic group. This amine group has the ability to accept a hydrogen atom, which makes it a basic group. Hence, proteins can be called as amphoteric in nature, that is, can act as both acid as well as a base. And D, 20 amino acids make up food protein, not 21. So the answer is option C. Hope this is clear. The entire amphoteric nature of amino acid is what makes protein behave both as an acid as well as a base. In case if you guys don't understand this message asking me to repeat the question, we'll repeat the entire process again. So question 3, which of the following correctly defines the characteristic of quaternary structure of proteins? A, linear sequence of amino acids. B, regular structural patterns such as alpha helices and beta sheets. C, the complete 3D network of different subunits. And D, 3D arrangement of atoms in the polypeptide chain.
Option C, ma'am.
Yeah, so option C belongs to the quaternary structure. So what are the other different kinds of structures of the protein, levels of structure of proteins?
Primary, secondary, tertiary, quaternary.
Yeah, so primary is the linear sequence of amino acids. So basically if you just arrange the entire sequence of the amino acids in a linear fashion, that constitutes the primary structure. And these amino acids, when they form specific structures such as alpha helix or beta sheets, they have a very regular shape. The hydrogen bonds will be formed between only certain regularly shaped regularly spaced residues. So alpha helix and beta sheets, all these kind of structures are termed as the secondary structure. So when multiple secondary structures come together and form a three-dimensional arrangement, that is known as a tertiary structure. And generally a tertiary structure is composed of one single chain. But in a quaternary structure, multiple chains will come together and this will form an interaction network. So quaternary structure is made up of multiple chains. So a linear sequence is the primary structure. The regular structural patterns such as alpha helices form the secondary structure. The 3D arrangement in a polypeptide chain, that is one single chain, is the tertiary structure. And multiple chains that complete the 3D interaction network is known as the quaternary structure. So the answer is option C. Moving to question 4. Denaturation of protein results in A, loss of its biological activity, B, decreased reactivity of constituent groups, C, increase in solubility, and D, resistance to enzymatic hydrolysis. So, any clue what is the answer?
Loss of its biological activity.
Yeah, so answer is loss of biological activity, but what about the rest of the three options? What happens when you have denaturation?
Reactivity increases.
Yeah, why does it increase? So what happens in a denaturation of a protein is that you will have a properly structured protein that will now lose its structure. And because it loses its structure, it will lose its biological activity as well, because the structure of the protein is extremely important for the function of the protein as well. So if the structure is lost, then the function of the protein is also lost. So when the protein unfolds, all the constituent groups that is present in the side chain, be it acidic group, basic group, or any hydrophilic or hydrophobic group, is now exposed to the solvent. And now since it is exposed to the solvent, the reactivity of the constituent groups also increases. So in a denatured protein, there is an increased reactivity of constituent groups. So why is the increase in solubility not seen? What is the reason for a decrease in solubility? So in a normal protein, when you look at, there will be something known as a protein core, that is the innermost region of the protein. This innermost region of the protein is made up of hydrophobic residues, that is, it has the ability to come together. Like say, even if you put oil in water, oil molecules have a tendency to come together because they are hydrophobic in nature. The same way, in a protein also, the protein core is highly rich in hydrophobic residues which come together and help it maintain its structure. Now, if there is a denaturation process, all the hydrophobic residues are now exposed to the solvent, which is not preferred. So what will happen is the hydrophobic residues of one protein will try to interact with the hydrophobic residues of the other protein. So multiple protein molecules, denatured protein molecules. So multiple denatured protein molecules will come together so that the hydrophobic residues are clustered. They do not like to interact with the solvent. So they prefer to be clustered. So all the hydrophobic residues of one protein will try to interact with the other protein, and hence it will start aggregating and result in a decrease in solubility. Is this understood? Since hydrophobic residues do not prefer to remain in the solvent, exposed to the solvent, they start interacting with other denatured proteins, causing aggregation and hence decreasing solubility. And what about resistance to enzymatic hydrolysis? Why is there no resistance to enzymatic hydrolysis? So the same thing, when a protein is when a protein has a very well defined structure, certain regions are not accessible for the enzyme to come and act. The moment you denature the protein and everything is now exposed to the solvent, the enzymes will now be easily to attack the protein because now it can capture the region where it can cleave off. So denaturation results in loss of the structure, which in turn affects the biological activity. The denature of the protein increases the reactivity of the constituent groups because now everything is exposed to the solvent. And because of this exposure, the hydrophobic amino acids, which were originally present in the core of the protein, will now be exposed, will try to interact with each other, resulting in aggregation and decrease in solubility. The last point is that the loss of the structure and the exposure of amino acids to the solvent increases the accessibility to the enzymes that hydrolyze the protein. Hence, denaturation increases the susceptibility to hydrolysis and does not give resistance. So the answer is option A. Hope this is clear. Moving to question 5. So we'll also discuss some numericals, some 1 or 2 numericals in this session regarding the microbial death kinetics. So the initial count of Clostridium botulinum in a food sample was 10 power 8 CFU per ml. So CFU means colony forming units. So 10 power 8 CFU per ml. After a heat treatment of 1.5 minutes at 121 degrees Celsius, the count gets reduced to 1 CFU per ml. Calculate the D value of Clostridium botulinum at 121 degrees Celsius. So how do you proceed with this? What does microbial death kinetics follow? What kind of a reaction does it follow? So microbial death kinetics follow a first order reaction rate. So first we'll discuss the given. So xt, that is x after 1.5 minutes, is 1 CFU per ml. X naught, that is initially that was present in the food sample, is 10 power 8 CFU per ml. Time is equal to 1.5 minutes and temperature is equal to 121 degrees Celsius. So when you're talking about microbial death kinetics, generally we consider it to be a first order reaction. That is, your rate is directly proportional to the amount of substrate. In this case, there's no substrate here. We're talking about the death of microbes. So in this case, it is directly proportional to the number of microorganisms present at that time. So dx by dt, which is the rate, is directly proportional to x. So if you remove the proportionality constant, you have minus k into x. So what is the significance of the negative here? Generally, if we remove a proportionality, we just add k. We don't add minus k. What is the significance of a minus k here? So the reason we add a negative sign is because we're talking about death kinetics, which means the amount of the cells are decreasing with respect to time. It is not increasing. So generally, if we look at any other chemical reaction, we'll add dp by dt. Let's say a product, the rate of product formation is equal to k into p or something of that sort. So in that case, it is increasing with respect to time, which is why you have a plus kt, plus kx. In this case, the microbes are decreasing, they are dying with respect to time, and hence you have a negative sign here. So dx by dt, which is the rate, is directly proportional to x. When you remove the proportionality constant, is equal to minus kx. So what happens when you integrate this equation from 0 to t? What is the equation that we get? So when you integrate from 0 to t, let's say x naught is your initial count of microbes and x is the count of microbes that is present at time t. So you bring x down here, dx by x from x naught to x is equal to minus k dt from 0 to t. So if you integrate this is ln of x. So ln of x minus ln of x naught can also be written as ln of x by x naught is equal to minus kt. So now if you take an exponent on both the sides, x is equal to x naught e power minus kt. Is this clear, the derivation? If you guys want me to repeat any step of it, you can let me know. So in this particular equation from the problem, we have x, which is 1 CFU per ml, x naught as 10 power 8 CFU per ml, and time is 1.5 minutes. So can you calculate and let me know what is k here? Is anybody calculating or should I move forward? Okay, so if we plug in the values in the equation, x was 1 CFU per ml, x naught was 10 power 8, and t was 1.5 minutes. So what is k here? So we take a logarithm on both the sides. Ln of 10 power minus 8 now will be equal to minus k into 1.5, which means k is 12.28 minute inverse. So we'll come to what is asked in the question. So it was asked, what is the d value of the microorganism? So what is the d value? How do you define a d value of a microorganism? Are you guys aware of this d value, z value and all?
Ma'am, decimal reduction of microorganisms.
Yeah, so what does it mean?
%90 of microorganisms are destroyed.
It is reduced by 1/10 of the fold. Yeah, so 90% is reduced, only 10% is remaining. Tenfold is wait. So the time required to reduce the number of microorganisms by tenfold. Yeah, so 90% is reduced and only 10% is remaining. So in this case, then how do you, with the k value, how do you figure out what is the d value? Now we know that d value, since it is the time required to reduce the number of microorganisms by tenfold, which means that now the current number of microorganisms that are surviving is 1/10 of what was originally present, which means x is equal to x naught divided by 10. Is this clear? So if you now plug in to the same equation, x is equal to x naught e power minus kt, since it is decimal reduction value, that is, you are decreasing the number of microorganisms by 1/10, this will be x naught divided by 10. And equal to x naught e power minus k d. Here, since d value is the time required, you can substitute t as d here. So you can cancel x naught on the LHS and RHS. So you'll have 1 by 10 equal to e power minus kd. So then you take a logarithm on both the sides. Ln of 1 by 10 is equal to minus kd. And hence d can be, the relation between d and k is that ln of 10 divided by k, which is 2.303 by k. So when you know the k value, you can always find out the d value or the other way around as well. So now we know what is the k value, which is 12.28 minute inverse. So what is the d value coming to be? You can substitute the k value that you obtained here in this equation. So this is a very this is a proper formula. You can use it in other problems as well. So d is equal to 2.303 divided by k, and when you substitute k as 12.28, you get 0.1875 minutes, and when you convert it into seconds by multiplying into 60, you get 11.25 seconds. The answer is option A. Is this method clear? Any doubts in the method, in the derivation? Any steps that needs to be repeated again? Okay, moving on. Question 6. Which of the following conditions can be used to control the Maillard reaction in foods? A. By keeping pH above the isoelectric pH of the amino acids, peptides and proteins. B. By keeping the moisture of the food to very low or very high levels. C. By decreasing the dilution in case of liquid foods. And D. By increasing very high temperatures. So what is a Maillard reaction? Have you heard of Maillard reaction?
Non-enzymatic browning.
Yeah, so Maillard reaction comes under non-enzymatic browning. And how does that happen?
Ma'am, the reaction between amine group from proteins and aldehyde from carbohydrate, it will
Yeah,
take place.
so the carbonyl groups of all the reducing sugars such as glucose and all will interact with the amine group in the amino acid. So if you guys remember the structure of the amino acid that I displayed in the second or third slide, that amine group will now interact with the carbonyl group of the reducing sugars, and that will form a compound called as glycosylamine. And these glycosylamine will further undergo multiple reactions to give a brown product that can form a brown color. So this brown color is imparted in foods, and this is known as the Maillard reaction. So how do you prevent Maillard reaction? Which of these statement is correct? So what happens to an amino acid? We'll go one by one. So what happens to the amino acid if we keep the pH below the pI of the amino acids? Do you guys understand what is a pI or an isoelectric pH? So what is defined by the isoelectric pH of amino acids? Okay, so
Net charge of the molecule is 0.
Yeah, so the net charge of the molecule at the isoelectric point is 0, which means that the amino group in the amino acid will be protonated and the carboxyl group will be deprotonated. So the carboxyl group will have a negative sign and the amino group will have a positive sign. So the net charge on the amino acid will be 0, and this happens only at the pH called as the isoelectric pH. So what happens when you decrease the pH below the pi or the isoelectric point? Which gets protonated and which gets deprotonated? So what is the characteristic point after very low pH? What happens in the solution? There's too much H plus ions, which is why you have a very low pH. When you have a lot of H plus ions in the solution, what happens to the amino acid? So when you maintain a pH below a pi where there is a lot of H plus ions in the solution, the H plus ions will be taken up by both the amino group as well as the carboxyl group. So when you have an H in the carboxyl group, that will form COOH, which means it will have no charge. But in case of amino group, when NH2 takes up a hydrogen ion, it becomes NH3 plus, which means that now the net charge of the protein is +1. Is this clear? I can show you the structure of the amino acid if you guys remember. So here in an acidic condition where there is too much of H plus ions, this will be COOH and this will be NH3 plus. So now the net charge of the molecule is +1. So what happens when the net charge of a molecule is +1? So when the net charge of the molecule is +1, the amino group was what was initially interacting with the carbonyl group of the reducing sugars. If now that is protonated, that will prevent the interaction with the carbonyl group. So if you have something else there in the amino group, it will not require it to form a bond with the carbonyl group at all. So this interferes, that is the protonated amino group in the amino acids will interfere with the carbonyl group, and hence by keeping the pH below the isoelectric point, you can prevent Maillard reaction. But if you keep the pH above the isoelectric point, the amino group will not be protonated and hence it can easily form this sugar amine compound. Is this clear? What happens when you keep the pH below the pi and above the pi? I am repeating here, below pi means amino group is protonated, which means no reaction between carbonyl group and amino group. Above the pi means the amino group will not be protonated and hence it can easily react with the carbonyl group and result in browning. So statement A is false. You have to lower the pH below the isoelectric point to protonate the amino group and hence control the Maillard reaction. So we'll go to B. What happens when you keep keep it very low, the moisture content of the food very low or very high? Why is the moisture content of the food very important in case of Maillard reaction? So if you guys remember the Maillard reaction, the sugar amine group will undergo a lot of condensation processes as well, which means it requires water molecules to complete the process of browning. So the water content in a protein plays a very important role in the browning reaction. So if you completely keep the food anhydrous, that is very low moisture content, it will not, the reaction will not proceed because now it does not have water to form that condensation, to proceed with the condensation reaction. That is, it needs to add water molecules to the functional group and then cause the browning reaction. If there is no water itself, the reaction will not proceed, and hence keeping the moisture content very low controls Maillard reaction. But what about keeping it very high? What happens when you keep the moisture content very high? So when there's very high water content in the food, everything will become very dilute. The same case, it's the same as option C as well. So when everything becomes very dilute, the reaction does not proceed because it requires a certain concentration of the substrate to proceed. If the concentration is very dilute, the reaction will not proceed and hence you can control the Maillard reaction. So by playing around with the moisture content of the food, it is possible to control the Maillard reaction. And D, what happens when you increase the very high temperatures? Why is Maillard reaction not controlled when you increase the temperature? So what is the major first induction of the Browning reaction? What induces the Browning reaction in foods? I'm talking about non-enzymatic.
Heat, ma'am.
Yeah, so the heating is the actual induction of the Browning reaction starting. So when you heat a compound, all your reducing sugars, the rings will start opening up. Only after the rings start opening up, you will have multiple reactions that proceed and form this melanin and other Browning compounds. So if you maintain a very high temperature, this will increase the ring opening process, and it will increase the Browning reaction as well. So you need to make sure that the food is kept at low temperatures. It is either very anhydrous or very dilute, and the pH is below the pI of the amino acids. That is, it is in an acidic environment. Only then you will be able to control the Maillard reaction. So to just summarize, keeping the pH below the pI of the amino acids results in the protonation of the amino group and hence prevents the formation of glycosylamine, which is the first step, and hence prevents browning. Decreasing the dilution increases the non-oxidative browning, and hence moisture of the food is kept either very low or very high to prevent browning. And increasing the temperature results in the ring opening of the reducing sugars and increases the browning reaction. So you need to make sure that the temperature is kept very low. So the answer is option B. Is this explanation clear? Okay, moving to question 7. The controlled Maillard browning reaction is desirable in which of these food compounds? A. Bread, B. Concentrated orange juice, C. Dried fruits and vegetables, and D. Canned food.
Bread.
Yeah, so because of the heat, when you bake the bread, because of the heat, the browning reaction will start, and people actually prefer the slight brown color in bread rather than the other food products. So consumers prefer this brown crust in bread, and hence the Maillard reaction is actually preferred or desirable in bread when compared to the other foods. The answer is option A. Moving to question 8. The reactive reducing sugars undergo which of these following reactions under heating in case of non-enzymatic browning? A. Ring opening and dehydration, B. Dehydration and enolization, C. Enolization and fragmentation, D. All of the above. So any clue what is the first step here? When you heat, what is the first step? We may discuss this in the previous slide. So the first step in a browning reaction is that the cyclic reducing sugars need to undergo a ring opening in the presence of heat. So the ring opening is the first step in the browning reaction. After that, what does happen? What happens? So on heating, they undergo ring opening, they undergo enolization, which is a kind of an isomerization process, dehydration as well as fragmentation. So So all these four reactions are majorly involved in the non-enzymatic browning processes. So the presence of reactive reducing sugars is what gives the color. So these undergo ring opening, enolization, which is a kind of an isomerization. The keto-enol isomerization, if you guys have heard of. So it undergoes this enolization or isomerization. Then it undergoes dehydration as well as fragmentation. So all these steps results in the formation of something known as melanoids or melanoidin. So all these compounds, melanoids or melanoidin, give this brown color to foods. Hence, all of these reactions are involved in non-enzymatic browning. So the answer is option D. So if you guys want a more detailed steps of these, it is mentioned in lecture 6, where an example of ketose sugar and normal glucose sugar is taken, and faculty has explained how it undergoes the formation of melanin. So a very detailed reaction mechanism is given. But these are the four major reactions involved in the entire process. So answer is option D. Moving to question 9, this is also a numerical problem. A food product is heated at 121 degrees Celsius for 25 minutes for achieving satisfactory cooking. The cooking process, however, destroyed vitamin C and it followed the first order kinetics. The D121 value for vitamin C destruction is 200 minutes. So calculate the percentage of vitamin C lost during the cooking process. So this is actually very similar to the previous numerical that we solved. So any clue how to proceed? So what can we find out from the D value? If you remember our previous numerical procedure. So D value is the time taken to reduce anything. It's not just microbes. Be it even vitamins or so, it is the time taken to reduce the amount of certain compound that you are looking at, be it vitamins or microbes, by a factor of 10. So from D value, what can you calculate? So from the D value, you can calculate the rate constant as we looked at before. So we'll first look at the given. So time given is 25 minutes. The cooking was done for 25 minutes. The D value is given as 200 minutes and the temperature was 121 degrees Celsius. So D, as we saw, is equal to 2.303 by K. The derivation of this is given in the previous numericals. So anyways, I'll be uploading this presentation and I hope it will be available in the NPTEL platform as well. So you can look at the derivation in that particular side. So the derivation, so D is equal to 2.303 by K. So if you substitute D value as 200 minutes, what is the K value that you're getting? So K is 0.011 minute inverse. We're keeping it as minute inverse. We'll finally calculate the time. Since time is also in minutes, we'll keep K in minute inverse as well. So K is equal to 2.303 divided by 200, which is equal to 0.011 minute inverse. So now you have the time, you have the rate constant. What can you figure out from these? How do you figure out the percentage of vitamin C lost? So it's the same equation since it's already given as it is the first order kinetics, and since we're again looking at the destruction of vitamin C, that is the concentration of vitamin C is reducing with respect to time, so it will have the same derivation as that we discussed in the previous problem. So from there we had derived that x is equal to x naught e power minus kt. So we have k here from the D value, we have time already given in the question. So x is equal to x naught e power minus 0.011 minute inverse into 25 minutes. So you have x by x naught as 0.75. So X here is the amount of vitamin C present now. It's not destroyed. X is the amount of vitamin C that is present now at time t. X naught is the amount of vitamin C that was present initially. So X by X naught will give you the amount of vitamin C that is left after 25 minutes. But we need to figure out how much was destroyed. So in order to calculate how much was destroyed, that is equal to 1 minus X by X naught. Is this clear? X by X naught is amount that is present. 1 minus X by X naught will give you the fraction that is lost. So when you substitute this value 1 minus 0.75 into 100, you have amount of vitamin C destroyed is 25%. So the answer is option C. Is this clear? So the first order reaction is used both for destruction of nutrient value in the food. It is also used for the destruction of microorganisms. It is generally follows the first order kinetics. Hope this is clear. Any doubts in this procedure? We can repeat again if you guys want. Okay. Moving to question 10. Pick the odd one out of the following. A. Sorbic acid, B. Peracetic acid, C. Benzoic acid, and D. Propionic acid. So what are these acids? Where is it involved? Any clue?
Preservative.
Yeah, these are preservatives. And how do they act? What is the point of adding an acid to a food? So adding acid will control the pH of the food. So why is pH a very important factor in the food process? So one of the major reasons of food spoilage is the presence of microorganisms. One is the Browning reaction that we discussed in the previous slides. The other one is the growth of microorganisms. So in order to inhibit the growth of microorganisms, people often add organic acids to the food to help control the pH. So what happens why is pH most important is that pH is involved in a lot of energy metabolism in bacterial cells. So bacteria produce ATP and other undergo energy metabolism reactions by the help of something known as the proton gradient. So you'll have a semi-permeable, you'll have a membrane, and you will have H plus ions accumulating in one side alone. So in one side of the membrane, you will have a high concentration of H plus ions. In the other side of the membrane, you will have very low concentrations of H plus ions. So there's a gradient across a membrane. So this gradient across the membrane is then utilized by the microorganisms to produce ATP. It also involves in the movement of the bacteria and all. So a high H plus gradient is actually involved in the energy metabolism in living cells. It's not just bacteria, it is also involved in human cells, in all organisms. So H plus ions are very, very important for the energy metabolism of microbes. So as you can see, the pH is extremely crucial for the organisms to survive. Apart from that, when you have a drastic change in the pH of your intracellular environment, all your enzymes, your proteins will be affected, because enzymes and all are very, very sensitive to the pH of the environment. So if you have a drastic change in the pH of the environment, your enzymatic activity will also drastically reduce. It will also involve in inactivation of certain enzymes that is crucial for the survival of microorganisms. So pH is not only involved in energy metabolism, it is also involved in the proper functioning of enzymes and proteins in the organism. So if you control the pH, you will be able to control the growth of microorganisms, which is why we add organic acids to foods. So which of these are are considered as preservatives by the food industries, and which is not used?
Peracetic acid.
Yeah, peracetic acid is not used as a preservative, whereas the rest of the organic acids are used as preservatives to control the microbial growth in foods. So answer is option B. So moving on to question 11, I think this is the last numerical in this session. So the D value for the destruction of Clostridium botulinum at 121.1 degrees Celsius is 5.7 seconds, which is 11 times of its D value at 131 degrees Celsius. What is the Z value of this organism and what will be its D value at 135 degrees Celsius? So we have discussed that D value is the time required to reduce the number of microorganisms by tenfold. So what is the Z value? Any clue what is the Z value here? So Z value is the temperature required to reduce the D value by 1/10. So if you plot a graph between D and temperature, the slope will be the Z value. This is understood? So if you had plotted so if you plotted x versus time, that would have given you the D value. If you plot the D versus temperature, that will give you the Z value. So Z value is the increase in temperature that is required to reduce the D value by 10. So your D final will be D initial divided by 10. Is this clear? What is the Z value of the organism? So the Z value will give you an idea of the thermal resistance of microorganisms. So what does it mean if the Z value is high for an organism? Does it mean is it resistant or not resistant? So if you have if a higher amount of temperature, let's say at 30 degrees my D value is somewhere around 10 seconds, but if at 40 degrees my D value is somewhere around at 1 second, then my Z value is 10 degrees. If my Z value is more, let's say some 20 degrees or 30 degrees, it means I have to drastically increase the temperature to reduce the D value, which means microorganisms are not dying if I just reduce it by 10 degrees. I have to increase it to 20 degrees or 30 degrees to kill the microorganisms, which means that higher the Z value, higher the resistance for the microorganisms. So Z value is an indicator for the thermal resistance of microorganisms. Higher the Z value, higher the resistance of microorganisms. So if you know the D value and the temperature, if you know the D value at a particular temperature and you know the D value at another temperature, you can calculate the Z value by this particular formula. So D at any other temperature is equal to D at reference temperature into 10 raised to minus T minus T ref, where T ref is the reference temperature divided by Z. So D ref is the reference D value. Here we already know that at 121 degrees Celsius it's 5.7 seconds. So let us consider in this particular question 121 degrees Celsius as your reference temperature. So if T ref is 121.1 degrees Celsius, then D ref at that particular temperature is 5.7 seconds. And it has been given that this is 11 times that of the D value at 131 degrees Celsius, which means that D at 121 degrees Celsius is equal to 11 times D at 131 degrees Celsius. Is this line clear? So D ref is equal to 11 times D is what is given in the question. So when you start substituting the values, you have here you will write it as D is equal to 11. D ref can be written as 11 times D. So that is basically 1 by 11 is equal to 10 power minus T is 131.1 degrees Celsius. This is where you have to find the Z value of this organism. So T is 131.1 degrees Celsius. T ref, that is for which we already know the D ref, that is 121.1 degrees Celsius, and Z value is what we need to find. Is this confusing or is it clear? It's very straightforward here. Your reference temperature will be 121 degrees Celsius. Your D reference will be 5.7 seconds, and that is said to be 11 times that of D. So if you just substitute the values here, you will get 1 by 11 is equal to 10 power minus 131.1 minus 121.1 divided by Z. So this is equal to minus 10. If you take a log of 1 by 11 is equal to minus 10 divided by Z. From this, you can calculate the Z value at a particular temperature. So, Z value of a particular organism. So, Z is 9.6 degrees Celsius. So, more the Z value, more the resistance of the microorganism. So, with this formula and with this Z, how do you calculate the D value at 135 degrees Celsius? We'll just do the exact reverse. So now we know the Z value. We already have the reference temperature as 121.1. We have the Dref as 5.7. We know the T that from where we need to calculate, which is 135 degrees Celsius. So all these parameters are known. We just have to calculate D. So we'll substitute this in the equation. D is equal to 5.7 into 10 power minus 135 minus 121.1 divided by 9.6. So this D value comes out to be 0.203. This is in seconds. So the answer is option A. Is this clear? So this is basically like a slope. If you have a D versus temperature in the semi-log scale, that is, there is a semi-log scale in the y-axis. So if you have D versus temperature, the slope will give you the Z value. That is exactly how this formula came. Your log scale in y-axis and T minus Tref will be your difference in x-axis. Z is the slope. So the answer is option A. So if you know the D and T are two different temperatures, you can always calculate the Z value. Any part that needs to be repeated? Okay, moving to question 12. The hydroxylation A in enzymatic browning results in A. Oxidation of diphenol to an orthoquinone, B. Ortho of hydroxylation of a phenol, C. Polyphenol oxidase of catechol activity, and D. Enzyme oxidations and polymerization of 5,6 spinato brown melanin pigments. So what are the different reactions that happens in an enzymatic browning? So there are two major reactions that happen because of the phenolase enzymes. The phenolase enzymes are the major responsible enzymes for enzymatic browning. So the phenolase catalyzes two types of reaction. One is the hydroxylation A, the other is known as oxidation B. So in hydroxylation A, you will have your orthohydroxylation of a phenol. As the name suggests, the phenol will undergo hydroxylation and then produce the browning compounds. Whereas in case of an oxidation process, you will have oxidation of the diphenol to an orthoquinone. So there are two kinds of reactions that happens, hydroxylation and oxidation, and according to that, you will have a different product that is forming, and ultimately this will lead to melanin pigments. So these are the two kinds of reactions that phenolase catalyzes, resulting in a browning reaction. So the answer for this, the hydroxylation causes the hydroxylation of phenol. It's very straightforward here. So the answer is option B. Okay, question 13. During a thermal processing, the temperature sensor is positioned at A, the point of the slowest heating in the container, where survival of microorganisms is higher. B, the point of slowest heating in the container, where survival of the microorganism is lowest. C, the point at which the rate of inactivation of the microorganism is higher. And D, the point at which the growth of the microorganism is higher. So, where do you generally keep the temperature sensor?
Option A.
Yeah, so you keep the temperature sensor at the point of slowest heating. So this generally, if you take like a cylindrical structure or so, the point of the slowest heating will be literally at the center, the bulk of the container. So there is where your heat will be given later. I mean, first it will start from the bottom. If you're heating from the bottom, it will start from the bottom, and the center is where you have the slowest point in the slowest point of heating, and that is where the survival of the microorganism is also higher. So, if you keep a temperature sensor at that region, you will definitely know that the points at the rest of the container will always be higher. The temperature at the rest of the container will always be higher, and the survival of the microorganism will be lesser in those regions as well. So accordingly, you can extrapolate as to what will be the survival of the microorganism in the entire container if you know the slowest point. So the temperature sensor is positioned at the point of the slowest heating in the container, where the survival of the microorganism is highest. The answer is option A. So from that point, you can extrapolate that how much is the container heating, how much are microorganisms dying according to the nature of the microorganisms present. So moving to question 14. Which of the following extrinsic factors affect the growth of the microorganisms in the food? A. Freeze concentration and oxidation potential. B. Oxidation and reduction potential. C. Equilibrium relative humidity and reduction potential. And D. Equilibrium relative humidity and temperature during storage. So what is the main characteristic feature of all the extrinsic factors that affect the growth of microorganisms? What do you mean by extrinsic factors? So what is an extrinsic factor?
The factor which affect the growth of microorganism externally.
Yeah, so it is not a characteristic of the food, it is not the characteristic of the microorganism, it is the characteristics of the environment, and it generally happens during the storage process. So once you've almost finished the processing of the food and you're trying to store the food, that is where majority of the extrinsic factors will come in effect. And this is the characteristic of the environment in which you're placing the food in. So freeze concentration, oxidation potential, reduction potential are all characteristics of the food. Only equilibrium relative humidity and the temperature during storage depend upon the environment where you are placing the food. So those constitute as the extrinsic factors, and the rest of the factors given here mentioned in this question are all intrinsic factors that depend upon the food, the kind of the food, the kind of constituents, the ingredients, the source. All those affect are the intrinsic factors, whereas the extrinsic factors come from the environment. So that is the equilibrium relative humidity and temperature during storage. So the answer is option D. So moving to question 15. Which of the following is the correct pair of enzymes that cause hydrolytic reactions? So what happens in a hydrolytic reaction? So in a hydrolytic reaction, generally we are looking at breakdown of a compound into fragments also. That's what generally happens in a hydrolytic reaction. So when you're talking about breaking down of compounds, which of these enzymes are important?
Option D.
Yeah. So lipases and proteases are the ones that break the major bonds. Lipases will break uh, all the cross-linking between the lipid molecules, and proteases will break the peptide bonds in the proteins. Whereas the other enzymes, that is ligases, are involved in what it is the opposite of hydrolytic reaction. It will in fact ligate two molecules, and hence that is how the name ligase comes into play. So ligases are not involved in hydrolytic reactions. They in fact help in the ligation of two different molecules. And lipooxygenase and peroxidases are all oxidating enzymes that result in free radicals and such. Only lipases and proteases break down the molecule into fragments. So lipases and proteases are involved in hydrolytic activity. Lipases involves in lipids, proteases involves in proteins. And since you have a breakdown of these compounds, this often results in quality loss of the food. So the answer is option D. Moving to question 16. Which of the following is the structural property of the protein? A. Emulsification, B. Coagulation, C. Cohesion, and D. Gelation. So all four of these are different processes that you see when you have protein in your food system. So which of these is related to the structural property of the protein?
Cohesion.
Yeah, so cohesion is involved in the structural property of the protein. What about the rest three? What property of the protein is related to the emulsification? So your proteins have multiple hydrophobic residues, it has multiple hydrophilic residues. So because of this mixture, it has both hydrophobic and hydrophilic residues. It has the ability to interact with both water and oil. So it has the ability to be present and stabilize an emulsion. That is why your surface properties, that is your hydrophilic residues or hydrophobic residues that are present on the surface of the protein, help in the emulsification or stabilization of emulsions. Hence, surface property of the protein is involved in emulsification. Coagulation is related to enzymatic properties. So in food industries, people often add enzymes to milk to form cheese, or like cheddar cheese and all are formed by addition of enzymes. These enzymes will help in the coagulation of the milk proteins and helps the formation of products such as cheese. So the enzymatic properties of the proteins are involved in coagulation or forming the coagulum or cheese. And degelation comes from the rheological properties of proteins. So some proteins have a property to come together and form a small region of very high concentration, and because of this very high concentration there's a lot of cross linking involved between proteins, and because of this interaction, that is the protein protein interaction is more preferred than protein solvent interaction. So because of this interprotein interaction, all of the molecules will come together, will increase the concentration, and the viscosity of that particular region increases. So this gelation process is related to the rheological properties of the protein. Only cohesion or elasticity, all those are involved in structural properties of the protein. Is this clear? Emulsification, because of the presence of both hydrophilic and hydrophobic residues in the protein. Coagulation is because you can add enzymes to milk and help the process of coagulation. Gelation, because gels are generally of highly viscous materials that contain both liquid as well as solid phases, and that will have a different rheological property when compared to your just liquid solution. And cohesion is a part of structural properties of the protein. So the answer is option C. So going to question 17. The attainment of elastic functional properties of protein is mainly due to A. Immobilization, B. Hydrogen bonding of water, C. Disulfide cross-linking, and D. Interfacial adsorption. So where does because of what does elastic properties of protein prevail?
Disulfide cross-linking.
Yeah, so the
Next
presence
week.
of disulfide cross-linking. So your proteins have residues such as cysteines or so. So these cysteines will have a sulfur group in their side chains. So this sulfur, one cysteine will form a disulfide, that is, it will undergo oxidation, and two of the sulfur molecules will come together to form something known as a disulfide linkage. So these disulfide linkages are present in proteins that have residues such as cysteine or in fact even methionine. But cysteines are what are majorly involved in disulfide cross-linking. So when you have two cysteines coming together, they often undergo oxidation in the side chains that cause disulfide cross-linking. So this contributes to the elastic functional property. So when you have a lot of disulfide linkages, your protein becomes very rigid. And if the disulfide linkages are reduced, or if you have if it is placed in a very reducing environment, these disulfide linkages are reduced and it forms a much more fluid kind of a protein. So the amount of disulfide cross-linking can tell us how rigid the protein is or how fluid the protein is. So this will give you an idea about the elastic properties of the protein. So what about immobilization?
During gelation.
Yeah, during gelation you will have immobilization. That is, multiple the proteins will come together. You will have protein-protein interaction happening, and because of this protein-protein interaction, there's a very viscous gel that is forming, and this will not allow the water to leak out of the system. So it will basically immobilize the liquid phase, and the proteins will kind of form the solid phase. So you'll have gels forming because of immobilization. So what will hydrogen bonding of water contribute to? What
Water
hap-
holding.
Sorry?
Water holding.
Yeah, water holding, or basically the solubility of a protein in water is because of these hydrogen bondings with water. That is, this protein solvent interaction is what helps something to dissolve. So the hydrogen bonding of the water will help in solubility and water absorption. And the interfacial adsorption is what contributes to the foaming processes. So if you guys remember the lecture videos, foaming mechanism happens because your partially denatured proteins or so will come to the air water interface, and because and at this air water interface, these partially unfolded proteins will form a film like structure, and because of this film you will have something known as air cells trapped in between, and these air cells will contribute to the foaming. So when the proteins come to the interface and then starts to unfold or so, it will form a film and cause foams. So interfacial absorption contributes to foaming functional properties of the protein. So I hope this is clear. The answer is option C, not option D here. Sorry. The disulfide cross-linking is what causes the elastic functional properties of the protein. So moving to the last question. Which of the following statement is true about the food gel? A. Polymeric networks are gels formed by globular proteins after heating and denaturation. B. Aggregated dispersions are gels formed by gelatin and polysaccharides such as agarose. C. Thermoplastic gels liquefy when heated. And D. Partial denaturation results in thermo-irreversible character. Which of these is true?
Option D.
Yeah, option D is true. What about A, B?
Polymeric networks are formed from gelation.
Yeah, so polymeric networks are formed when you have gelatin and polysaccharides such as agarose coming together and forming a network. And aggregated dispersions are formed by globular proteins. So it is the opposite. Polymeric networks are formed by polysaccharides. Aggregated dispersions are formed by globular proteins after heating and denaturation. So what's a thermoplastic gel then?
They doesn't liquefy when heated.
Yeah. So they become soft upon heating um, instead of being liquefied. So if you heat agarose and all, it will start to liquefy. On the other hand, thermoplastic gels will soften upon heating. Something like aloe vera gels or so, or starch-based gels will liquefy will soften upon heating instead of liquefaction. So thermoplastic gels do not liquefy when heating; they soften when heated, and partial denaturation results in thermo-irreversible character of certain gels. And thermoplastic generally means as thermo-irreversible. That is, you cannot just go back to its original content, original constituents where it was not a gel. Partial denaturation results in thermoplastic or thermo-irreversible character of the gels. So answer is option D. So with this, we conclude the week 2 live session. If you guys have any doubts in the questions that were discussed today, please let me know.